Coupling analysis of single speckle imaging and traction force microscopy can analyze the molecular mechanics for Grosscon Advance and navigation. As the techniques use commercially available materials and standard microscopes, researchers can readily adapt the techniques in their studies.
Begin by treating the neurons with tetramethylrhodamine or TMR ligand at a dilution of 1 to 2000 in culture medium on the day in vitro 3. Then, maintain the neurons at 37 degrees Celsius with 5% carbon dioxide for 1 hour. After incubation, wash the TMR ligand 3 times with pre-warmed PBS. Remove the PBS before adding 0.5 milliliters of warmed Leibovitz's L15 medium into the neurons. Maintain the neurons at 37 degrees Celsius for 1 hour. Next, turn on an epifluorescence microscope and set the stage-top incubator to 37 degrees Celsius. Then, place the TMR ligand-treated neurons in the glass-bottom dish on the warmed stage-top incubator. Set the image acquisition parameters, such as exposure time at 500 milliseconds for LifeAct and HaloTag Actin fluorescence channels, and binning as 0.065 micron by 0.065 micron per pixel at time interval of 3 seconds for 50 frames. After selecting a growth cone that strongly expresses LifeAct and weakly expresses HaloTag Actin, close the field on the diaphragm.